Lucas. Good morning, everybody. My name is Greg Dutton. I'm a Forensic Science R&D Program Manager at NIJ, so you're going to see several of us come up and introduce the speakers throughout the day. We're really happy to have the audience in the room and also online. We have a significant online audience. We're going to have speakers coming up. They have 20 minutes to speak, and then we'll have a 5-minute question and answer period afterwards. So in the room, we will have a mic that can be passed to you if you have a question. Online, you can enter questions online, and we will bring those up at the end of the talks. We're happy to host this here today. This is just a look at a small portion of our forensic science R&D portfolio at NIJ. We have over 100 active projects at any time. We also encourage you to visit the poster session at the end of the day. So at 5 p.m., we have a poster session just down the hall with lots more of our projects to look at. And also, of course, throughout the week at the AFS main meeting, you're going to see lots of NIJ-supported research, so look for that as well. So the first 4 talks are going to be from my portfolio, Physics and Pattern and Trace. We traditionally bring this first thing in the morning because you've just had breakfast. You can put your full attention to it. It's like taking math or chemistry at 8 a.m. So first speaker today is Alan Zheng. You can see the program for his full bio, but Alan is a mechanical engineer at NIST, and he has played a key role in applying 3D imaging to firearms forensics. So I'll turn it over to Alan.
Thanks. Hello, everyone. This is the first speaker. That's awesome. So today I want to talk to you guys about a research that we conducted with support from NIJ, looking at population statistics for firearms and toolmark analysis. Typical disclaimers, I will be mentioning commercial products. Doesn't mean any kind of endorsement from NIST. And this project was, again, funded by the NIJ. So why are we doing this? One of the things that we started to look at in firearms and toolmarks is that there's been a lot of paradigm-shifting technologies being introduced. We're now able to measure 3D surfaces. We're able to come up with algorithms to start scoring and being able to assign quantitative similarity metrics to toolmarks, something that the NAS report, PCAST report, they're all looking for. They want more objectivity. So once we've been able to develop these algorithms and being able to score things, now we need to look at how do we put that score into context. Now, DNA did this early on when they started sampling different races to look at the probabilities. That's pretty much what we need to do for firearms as well. The tricky thing about firearms is it's not a closed system, meaning there's almost an infinite possibility of different types of tool marks. There's no neat statistic you can do. The only thing you really can do is empirically sample a lot of known matching samples and a lot of known non-matching samples to really see when you have a ground truth comparison between 2 known matches, what is the probability of getting that score? And the same thing for known non-matches. So this project, we wanted to tackle 4 major popular brands of firearms within the field. You can see we're looking at Glocks, Rugers, Smith & Wessons, and Sig Sauers. And as part of this project, I leveraged my relationships and help from forensic laboratories across the United States and asked them to just shoot their reference collection for me. A lot of major labs had hundreds of firearms in their collection. I sent them ammunition. As part of this study, we made sure that the ammunition is exactly the same across the board for everyone. So we removed that portion of removed that as a variable. And our target goal was to get at least 100 firearms within each of these populations so we can really study how the different populations will look. And the algorithm we used here is something that NIST came up with, this congruent matching cells method. And to put simply, when you have a known matching samples, you have a lot of CMCs. The more CMCs, the better. And all it really is doing is looking at small, localized sections across the breech face of a cartridge case here, and searches across a compared surface and finds its best matching location. When you have a group of them in congruent orientation and location, then you have what's known as a CMC. So here's an example of a known matching comparison. In a known non-matching comparison, you would expect very low CMCs. So these cells will be all over the place. There's no congruency. So this is how we're scoring it. Lower the CMCs, the worse similarity. Higher CMCs, the better similarity. So then we want to look at the statistics. And one of the easiest ways to look at it is let's just plot the histograms. What should known matching comparisons look like? On this graph here, you can see on the x-axis is the comparison score. So you would expect all your known matching comparisons to be scoring very high, all your known non-matching comparisons to be scoring very low. Y-axis is just the probability of achieving those scores. In a perfect world, everything is separated. We had no errors. But in the real world, we do have crossover between these two distributions. Anytime you have a crossover of these distributions, you end up with an error. In this particular study, we wanted to study the false positive and false negative error rates of the NIST system in our statistical models here. So we set a threshold of CMC equals to 6. So anytime a CMC score goes above 6, we consider that a matching comparison. Anytime it goes below 6, we consider a no non-matching comparison. So you can see if a no non-matching tool mark scores above 6, you end up with a false positive. Anytime a no matching comparison goes below 6, you have a false negative error rate. And those red and blue triangles there is the error rate that we're trying to calculate. So here's a quick preview of the 4 different populations and their no matching and no non-matching comparisons. So anytime you see a red distribution or red histograms, those are no matching comparisons. Blues are the no non-matching. And within this study, in every population, we calculated all the available no matching comparisons. So for example, Glocks has 166 no matching scores. Ruger's have 180. Smith & Wesson has 139. SIG has 127. And for each population, we also calculated a total of 10,000 no non-matching comparisons. So you can see these graphs, and one of the things I want to draw your attention to is how tight that blue distribution is. So when it's a no non-matching comparison, the CMC score is very, very low, and it's consistently low. And this is very good for us when we start looking at the statistics. Now, the no matching comparison does tend to change from population to population. And this is typically mirrors what an examiner's experiences as well. Certain populations of firearms mark better, certain firearms don't mark as well, and you don't have as much of a distinction. So let's go through each of these populations real quick, and you can see the next series of slides. I broke each population up into the full population, you can see here, and the false positive error rate, as well as the false negative error rate. And that green line, again, is CMC equals to 6. So within each population, we can also separate them by different class characteristics. So here's the Ruger full population again. And in the summary table, we'll see the different ones. So you can see Ruger's, the no matching comparison starts to trend a little bit to the left. But again, the false positive probability is still low. Smith & Wesson, and 6 Hour. 6 Hour actually was one of the ones that had the highest false negative probabilities. And 6 have been known to not mark very well. Very smooth surfaces on the breech face, which tends to score very low for no matches for quite a good number of them. And one of the things I tell examiners all the time, that algorithms are not some sort of black magic thing that can immediately see things that you can't see. So anytime you don't see individual characteristics on a surface, algorithms not going to just magically pick them out either. And we're seeing a lot of correspondence between examiner experience and the algorithms as well. So here's a summary table. And again, we broke each population up with granular breech faces and parallel breech faces. These are observed class characteristics within each of the firearm populations as well. You can see the false negative error rates down the right, with the lowest at 1.62% for Smith & Wesson for parallel surfaces, and the highest for SIG granular, which essentially just means a very smooth breech face. But you'll notice the false negative error rate is extremely low all the way down the line. And this means we may be able to do further analysis here to really pull out what does this really low false positive error rate mean, and how can it be advantageous when we're looking at different populations. We did the same thing for firing pins as well, for all of these firearms that we tested. You see kind of similar results, and SIG being the problem child again. SIG firing pins are extremely smooth, meaning when we really can't pick any kind of details out of them. You can see a lot of that known matching comparisons score very low as well. We can do the same kind of table analysis, but again, you still have very low false positive error rates. And keep in mind, examiner performance on false positive error rates is close to 1.5%. So the algorithm is in line with what examiners typically see, and when there's no non-match, it scores very low. So then we can also look at, if we only care about the known non-matching comparisons, so let's look at random match probability here. At what point in each of these populations do you get 1 in 10 million chance of a false positive scoring that high? And in each of these populations, as CMC goes to 8, you've already reached that point. And that 8 is actually a pretty low score. Remember, our threshold is at 6. And you can see the scores down there go all the way up to 25, 30. So it doesn't take very much to have a very, very low probability of randomly matching a false positive here. We see the same thing for Smith & Wesson and SIGs as well. And these actually scored a little bit better at 6 here. So the last thing we wanted to do is look at how do these populations and these distributions work in a score-based likelihood ratio paradigm here. Because we really need to look at both sides of the coin. What I just talked about, 1 in 10 million, that's great. But if you have the distributions on the right there where all your known non-matches, all your known matches score very low, like we've seen in the 6 hours, that 1 in 10 million doesn't really mean very much. So what we did was we calculated score-based likelihood ratios for all of these different populations. And a quick primer for a score-based likelihood ratio is for any given score, in this example S, you look at the probability of achieving S, given that it came from known matching comparisons, divided by the probability of achieving that score of S, given that it came from known non-matching populations. And score-based likelihood ratios goes anywhere from 0 to infinity. The higher the score, the more support for the prosecution's hypothesis. And we notice quickly, if we just look at our threshold of CMC go to 6, the score-based likelihood ratios are not a fixed value, and they do vary a little bit. You see from population to population, the number does change. However, one thing we notice is they're within the same order of magnitude, which is good. And if you really look at all the populations stacked on top of each other, again, the known non-matching population for all 4 of these different manufacturers of firearms are essentially the same distribution for known non-matches. Known matches do vary a little bit, but not by very much. Now here's a graph that we started to use, where on the x-axis you see the score of CMC, and the y-axis is a log of the likelihood ratio at each of those given scores. Because once we have the distribution, we can calculate the likelihood ratio for any of these CMC scores that you achieve. And this is what we use for reference populations in the future when we calculate any score that you achieve from the evidence themselves. And what we did was, in that red plot, we combined all 4 populations together, meaning we don't care about the brand, we don't care about anything else. We just combine all 4 populations together and compare that with each of the individual populations. And what you see is, with the exception of Glock, Ruger, Smith & Wesson, and SIG, they're right on top of that all population. What this means is you could, theoretically, merge these 3 different populations of firearms together to create your reference population and not have a dramatic difference in your reported score-based likelihood ratios. Glock is a little bit different, but not by much. Everything's still within an order of magnitude. So this is very good for us when it comes to building a national database of these types of distributions of how many firearms we need. We see similar results for firing pins. And again, notice that these plots only go up to about CMC equals to 8 before they plateau out. And the typical question I get is, why is there a plateau? We utilize something that was developed at the NFI called the ELOB limit. It's empirical lower upper bound limit. because likelihood ratios, as we all know, goes to astronomical values that's unsupportable in the tails. So this method allows it to limit how high the likelihood ratio can go. So in all four of these populations, at about CMC equals to 7 or 8, you already hit the highest limit for this population. The more calculations we do for known non-matches, the higher that limit goes. We also looked at the different surface materials. This is something that Dr. Martin Biker at NFI did. And you can see, doesn't matter if it's a nickel primer or a brass primer, the score-based likelihood ratio is extremely the same. Same thing for firing pins as well. And how does this work? Well, remember, those known non-matching distributions are extremely tight, they're extremely low, and they dominate this likelihood ratio the higher that score goes. So this is really good for us in a sense that we're very confident that known non-matches will score very low. So what we've learned from this study is that the CMC algorithm yields really low false positive error rates across pretty much any population, any kind of class characteristics. And this is going to help us in the future when we start combining different populations together in a reference population. We still need to build this reference collection, so a lot of the lessons we learned here is being applied to the reference population database of firearm tool marks, which we're collaborating with the FBI to build. This will be a national reference population, and it will be the basis of any kind of score-based likelihood ratios for firearms and tool marks moving forward in the future. But we still do need to research all the different combinations of class characteristics, making sure that we can defend why we combine different populations together and whether it's a valid thing to do or not. And that's something that we'll continue to do as we start building the reference populations. So I'd like to thank all the participating laboratories who provided samples. And again, thank the NIJ for providing funding for this research. It's been instrumental for us moving forward in our current research as well, to help understand how the different populations of firearms and tool mark and their statistics play out. So with that, I'll take any questions. Thank you. Is there another manufacturer that might have similar characteristics like a Glock, the smoother firing pin, like you mentioned?
Let's do some Groundhog Day. That works. My question was, for future studies, are there additional firearms manufacturers that may have characteristics or behave similar to the 6-hour class?
Yeah, and one of the things that makes, again, firearms tricky is manufacturers are always changing how they make things. And when you build these reference populations, it comes with a whole set of assumptions. And when you have a brand new manufacturing technique, and this is why we rely on the examiner population and the community, is we need to sample some of those, making sure that this new way of making firing pins, a new way of making breech faces, still falls within our current models. If they don't, then we need to make a new population for them. So this is going to be an active, ongoing process where we're going to have to keep track of these new things coming out. And we may not be able to use the system for certain populations of firearms because we haven't sampled them enough. We haven't validated that enough. So it's an incremental step process.
Mm-hmm. In every case, it looks like your threshold was set so high that there weren't actually any false negatives, and yet your false negative rate was always non-zero. Was there always an actual false negative above,
The false positives. False negative error rate was extremely low, and mainly because we didn't see any CMCs go more than 4, and we maybe only had two occurrences in a sample size of 40,000 comparisons where it was that high. we set a CMC equals to 6 as a more conservative criteria, and it's almost, and we did 6 because it was a parallel to consecutively matching stria in firearms and tool marks. So we set it as 6, see if it works. Our number one goal, obviously, is never to have any false positives. And with that 6, even outside of this study, we've never achieved anything higher than that.
Thank you.
Are you still building your database? There's someone that wants to offer assistance with their reference collection if needed.
I have a laboratory probably of thousands of cartridge cases and bullets that is just sitting there waiting to get measured. So as of this point, we're no longer collecting just samples from the wild. There was about 3 years of my life where I went around begging for samples, and the community were really helpful. So as of right now, our main focus is building the reference population with the FBI, and they're going through their entire collection, test firing every single one of their guns with 11 different types of ammunition to build this reference population. So it's a monumental effort that we're focusing all our efforts on right now. And by the way, all of the raw data that we collected from this study we'll be posting on the NIST research database. So it's open access for anyone to test what we did and maybe try your own methods as well.
Alan has clear relevance to practice, but it actually is really interesting from a research perspective. And I don't know whether you all are considering looking at some research questions that have been relevant to the field using this approach. Like one of the things that I have in mind is like, how much is the shot-to-shot variability within a particular firearm,
right,
or how persistent really are, like I've seen claims that marks on bullets and cartridge casings aren't that persistent over 5-10 years, and that's the reason why you might see variation in reanalysis. But this technique would be a great way to kind of look at interesting questions like that from a research perspective too. Are you all looking at anything like that?
Yeah, we do have populations on the NIST research database where we looked at firearm decay from shot 1 to shot 10,000, and we've looked at shot-to-shot variability as well. The biggest issue is from a practicality standpoint. A forensic lab is not going to test fire that firearm that they recovered 100 times just to build a population of how much variation you see within this firearm. So a lot of what we do is we try to account for some of those variabilities, and this is a whole different talk, and I think I'm out of time, but one of the things we can do for likely ratios is to be extremely conservative. Everything we do is extremely conservative because we know there's a lot of variability in a lot of these other questions that could affect what your likely ratio is, but we can talk offline about that as well. All right, thank you.
Hey, Alan. Sorry, Alan.
One more.
Yeah.
Okay.
I was curious. You were saying that you were combining, I believe, the Glocks and another brand of manufacturing based on the behavior of the likelihood ratios.
Say that again.
When you were saying it looked like you could pool two different populations of firearms
Yes.
together, was that based on the performance curves of the likelihood ratios, or was it the known same match distributions and the denominator distributions for known non-match distributions?
So what we wanted to test to see is if we combine different populations together, is there a major effect on the likely ratios reported? So we made a more generic population versus very specific populations. That's all.
Thank you.
Yep, thanks.
All right, thank you, Alan. We're actually a little bit ahead of time, so plenty of time. Our second speaker in this session is Tatiana Trejos. She is Associate Professor in the Department of Forensic and Investigative Sciences at West Virginia University. Tatiana may be one of the busiest PIs in NIJ's portfolio. She has so many projects recently, so many students. Here she's going to talk about fracture fit, which I think is a great bridge between the physics and pattern and trace. Tatiana?
Thank you, Greg. How do I pass the slides? Good morning, everyone, and thanks for the opportunity to discuss our research at West Virginia University assessing the strength of trace evidence fracture fits. So this project was sponsored by NIJ, so the opinions, findings, and conclusions are those of the authors and do not necessarily represent those of the Department of Justice. So the main reason why physical fits are considered a valuable evidence is because when we take a physical fit, an item exhibits physical features that demonstrate that they were once part of the same object. And that is considered the highest degree of association that we can achieve in physical fits or trace evidence. In addition to that, physical fits are very convincing and easy to demonstrate to a jury. So in order to illustrate that, I asked my colleague Troy Ernst at the Michigan State Police to share with me one of the many examples of physical fits that he has done to illustrate this aspect. So in this particular case, this is a hit and run that happened in 2019, and the only evidence that was left near the victim was what appears to be pieces of an automobile vehicle and apparent pieces of automobile paint. So in this kind of examinations, usually trace evidence is the only material that is left that can provide a link to a particular perpetrator. So in these particular cases, the main step is to do chemical and physical analysis of the paint to determine if it's automobile paint and if it is original equipment, OEM paint. So in this particular case, Troy did this analysis, determined that this is an OEM paint, and therefore was able to do a PDQ database search and provide investigators with potential hits of year, make, and model of vehicles. So with that, they were able to find a potential suspect vehicle, and as you can see in the images here, there is substantial damage on the hood of that vehicle. So the first step in this case, type of cases, where we suspect of a potential physical fits, is do a macroscopic examination and try to align the physically the evidence, the question evidence, to the vehicle in this case. And as you can see, there were like good alignment of the fracture of the edges observed here at the macroscopic level. The second step, once a potential physical fit is established, is to look at more deep detail at a microscopic level. So this is what you are seeing here. This is a microscopic observation of that fracture fit. We have the top and the bottom part of the paint. So not only you can confirm and see that there is a nice alignment between the two items, but also there is a correspondent of microscopic features. And in the bottom part, if we change a little bit the magnification and the lighting, you can see that we have a lot of striations in that bottom part of the paint that go in many different directions and align very nicely from one piece to the other. So in this particular case, Troy was able to find not one, but 3 physical fits. So in his opinion was that these 3 paint chips originated from the hood of the vehicle. And the alignment of fracture edges displayed between the question paint chips and the hood paint will not be expected except but paint that was once formally connected. And this is what we call a fracture match or a physical fit. So you may be wondering then, with such a convincing evidence, why we will be spending so much time in doing additional research. And the reason for that is that not always physical fit, arriving to a physical fit conclusion is that straightforward. And even in this type of cases, the scientists need to have a substantial scientific basis for forming their opinions. So the steps that we followed to determine what were the gaps for the research in physical fit examinations. First of all, we have to understand who are our customers. Judges, the jury, lawyers, and the criminal justice in general. But most importantly, what are the expectations? In the courtroom, the expectations are not only to be able to find and demonstrate something just visually like we saw that is self-convincing, but being able to support the scientific validity on how we arrive to those conclusions. So with that in mind, we identified 3 main areas in which the physical fit examinations can benefit: objectivity, consensus, and scientific reliability. So as you can imagine, human judgment is very critical in this type of examinations. And although subjectivity is not always a bad thing, it can be risky if we don't have standardized criteria. We also were in need in developing consensus of what constitutes an individual characteristic, how that may change between different materials, and when is good enough to call something a physical fit. We also wanted to evaluate in more detail what are the error rates in this field and what are the factors that can affect the error rates in this type of examinations. So the way that we approach this type of research is that we work very closely with our practitioners. We created multidisciplinary collaborations between scientists, statisticians, and human factors to provide different perspectives to the experimental design. We developed standardized comparison criteria that I'm going to be presenting today and evaluated the error rates and applicability in the field. So we focus on 3 main types of materials that are heavily submitted to crime laboratories: duct tapes, textiles, and automobile plastics. And for each of those materials, the overall goal was to arrive to standardized, systematic, reproducible, and quantifiable comparison criteria. So for each of these materials, we had 3 main stages in the research. The first one was to establish the criteria and the terminology of what constitute individual characteristics. Then develop a systematic method and quantifiable metrics to evaluate the quality of a fit. And finally, design a large population data set that represent casework that we receive and evaluate it through interlaboratory studies to assess the error rates. In these processes, we did a lot of different statistical evaluation, but the end product is a very simple and adoptable method. So our database corresponds to more than 9,000 physical samples of duct tapes, fabrics, and polymer plastics, and more than 4 times that digital database of images that we use for our computational algorithms to do also some comparison and compare that to a human. So in each of these materials, we follow the typical workflow that is proposed for physical fit examinations, and this ASTM standard just recently was accepted, but it was a work of about 3-4 years from the OSAC Physical Fit Task Group. And the physical fit examination can be explained through this workflow and usually contain 3 major steps. The first step is the assessment of the general features and to determine whether or not the materials are suitable for physical fit examination. The second step consists in evaluating the features and compare those features at the macroscopic level, and finally, compare those features at a microscopic level. So what was new in our approach is that we incorporated in each of these steps a standardized terminology and criteria to do the examinations by material, as well as systematic and demonstrable documentation of the thought process in each of these stages. So these are how these 3 stages look like for duct tape, for example. The first step is to look at the general class characteristics of the known sample first, and then the question sample, and then observe and report the potential overall alignment that we observe between the 2 compared items. The second step consists in doing the macro assessments. So for duct tapes, we split the width of the tape into 5 equal sizes, and then we evaluate a series of features that I'm going to be describing in a moment and document that. And finally, we move to the microscopic stage. In the microscopic stage, the main important feature was to determine how small was the minimum comparison area for each material. So for duct tapes, that was relatively simple because we decided to use the minimum comparison bin as each area between the screen line areas, as you can see here, the fabric in white. And the reason for that is because the screen has been demonstrated in the past to be very, very consistent across a single roll of tape. So we consider that area as the minimum bean of comparison, and for each of those beans, we independently evaluate all the features that we are going to describe in a moment, and determine whether or not that bean has support for a fit, in which case the examiners will input a 1, a non-fit, in which the examiner will input a 0, or inconclusive, in which case we're going to input a 0.5. At the end, the template will automatically add up all those numbers and divide that by the total number of comparison beans, and that will provide a score that represents the quality of that physical fit. This method is quite simple, but provides a systematic, quantifiable, reproducible method that is also very easy for peer review and verification process. These 3 steps are recorded and documented in a very simple Excel template that has auto-populated cells, so the examiner can be easily computing all the thought process. And the second and the third step that I mentioned contain the documentation of 9 features. Some of these examples, the first feature is the alignment of the edge pattern morphology. So in this case, as you can see here, we can evaluate that at the macro and microscopic level and determine whether or not that alignment is indicative of a fit or a non-fit. We also look at the alignment of severe dimples that might be present or not, depending on the manufacturing process. And when present, they can be indicative of a fit if they align, like the one that is highlighted in green, or indicative of a non-fit if they misaligned. I won't have time to cover all the different features, but we came up with 8 different additional features that provide distinctive characteristics and help us to determine that fit and non-fit determination in every bean. Then in that third step, this is an example of the template in which the examiner will document for every bean all these features and evaluate whether or not it's indicative of a fit or fit in each bean. And when they input at 1, that will be color-coded with green. Inconclusive areas will be color-coded with yellow, and the non-fit areas will be colored with red. At the end, also, the template will automatically calculate that edge similarity score. And the advantage of this simple color coding cell is that you can see at a glance where are the matching and non-matching areas. And when you are doing a peer review process, a verification process, you can easily compare that in a few seconds and evaluate what are the areas of discrepancy, and then go to the template and look in more detail what was the whole thought process for the examiner. So this is an example of how that color-coded regions look like for a fit that has a score of 91%, an inconclusive of 51%, and a non-fit. So with that method, then we evaluated a full population data set that represented typical casework. And the factors that we evaluated for duct tape was the quality, high quality, medium quality, or low quality, the separation method, hand-torn or scissor cut, and whether or not the samples were heavily stretched. And then we evaluated how these factors may affect the error rates. So this figure here represents all the thousands of samples that we evaluated for physical fit examinations. So bear with me as I explain the content of this graph. So in the bottom, in the y-axis, in the x-axis, you will see the edge similarity score that was reported for each examination, and that can go from 0 to 100. From top to bottom, in the left-hand side, you will see 9 data sets. The first 3 ones are low quality, the next ones are medium quality, and the bottom ones are high quality sets. And for each of these sets, from top to bottom, we have scissor cut, hand-torn, and hand-torn stretch. And for each of those data sets, we have true non-fits on the top and true fit distributions on the bottom. So as an example, a couple of things that we learned from this is that most of the data sets, regardless of the factors that we were evaluating, had a very good separation of the true fits and true non-fit scores, with true non-fits or scores less than 40 supporting a true non-fit and scores greater than 80 supporting a true fit. The box plots distribution shows the distribution of those scores in each of the population, and the one that had the most variability in the true fits were the high quality when they were hand-torn. And the reason for that is that when these high-quality tapes are hand-torn, they tend to leave very few distinctive features. And those scores for the true fits tend to be more widely distributed. So we can use that information to look at the performance rates. In this case, we can see that we observe very high true negatives and true positive rates for all the data sets. Most importantly, for the thousands of samples that were evaluated, we didn't find a single false positive, which is one of the main important assumptions that we have made for many years in physical fit examinations. And with the exception of the ones that I mentioned, the high-quality hand-torn that had more false negative rates, the accuracies were very good. In addition to that, when we provided a blind subset to 2 independent examiners, the results were almost identical. Just like Alan mentioned, we can also use these scores to calculate the score likelihood ratios, and that provide another means to estimate the probative value of the evidence. So the next step was to evaluate that through interlaboratory studies. So we provide a very quick training to willing participants. We had 38 different participants, and we submitted 3 different kits for the examination. These correspond to 3 samples that were non-fits with low scores, 3 samples that were fits with high scores, and 1 sample that also was a true fit that had the score below that threshold of 80. What we found, again, is that we didn't have any false positives reported and high accuracy in the determinations. Most importantly, we also observed for all the different non-fits and the fits that were challenging and the ones that have higher score, a very high consistency among all the participants across all the different kits that were analyzed. So with that thought process, we also follow a similar type of research for textiles, and the factors here are a little bit different. So we evaluated 2 separation methods, torn and stab, different compositions of fabrics, different constructions, including knit and weave, and different designs, single color versus multicolors. For textiles, we defined which were the 8 features that provided the most individualizing characteristics and included that in the reporting template. And this table summarized the performance rates for the stab pairs in yellow and the hand-torn pairs in blue. So as you can see, we also observed very high accuracy in these examinations. But unlike the dog tape, false positives were very low, but were not 0. So that represents an important warning in our community that not every single type of material is feasible for physical fit examination, and there are certain factors that can influence the error rates. However, in general terms, as we look again at the distribution of those error rates on the samples, we can see that there is a very clear separation in the scores received by true non-fits and true fit samples, with most variability observed in the knit samples, hand-torn. The interlaboratory study was also conducted and was included a fit of high score, a fit of lower score, and non-fit. And these ones correspond to digital images that they were also able to observe a high magnification. And again, we observed very good consistency on the scores and the reports by all the participants. For automobile polymers, we focus on headlamps and bumpers that are very commonly encountered in hit and runs and other type of accidents. And for these materials, we came up with 10 distinctive features that we want to document, just as I mentioned earlier in the templates. But the difference in this data set is that we included the feedback from participants from the interlaboratory studies, in which they came to us and said, it will be wonderful if we can include not only the qualitative descriptors of all the features that are important, and then the numeric metric, but if we can document how much weight we put in each of those features while we are doing the observations. So we came up with a very simple scale that goes from 2 to -2, depending on how much that supports a fit and a non-fit, and we call that the feature prominence sum. The accuracy in this particular case, again, was fairly good. In these hundreds of samples of polymers, we didn't find a single false positive, just like in duct tape. but the false negatives were a little bit more prominent because of distortion that may happen during the break-in event that may hide some of those. But as we can see here, the edge similarity score has a fantastic separation of true fits and non-fits, and the FPS value provide an additional information of that thought process. And what we can see here in the graph that you have on your right-hand side is, or your left-hand side, I'm sorry, is that values of less than -5 strongly support a non-fit. Values greater than 15 strongly support a fit, and something in between are typical of the inconclusive results. So in summary, the studies provides further scientific foundations through performance rates from large population studies, interlaboratory testing, statistical analysis, computational models that I didn't have the time to explain, but if you are interested, please reach out to me. And a big database that now is shared through the NIJ that correspond to physical and digital samples. This led to a simple and cost-effective solution to improve the way that we can document physical fit examinations. And what the method can provide is a simple approach for physical fit comparisons that can complement and modernize current practice, that provide a systematic method to facilitate standardization during that comparison process, provide data-driven conclusions that inform the opinion of the examiner, and allows a transparent documentation that is helpful not only for presentation of our evidence in the quorum, but also through verification and peer review process. The qualitative descriptors and criteria, as well as the quantitative methods, have demonstrated to provide a strong scientific validity to physical fit examinations. So in the future, we are interested in continue evaluating these methods and factors that can affect the error rates and how we can disseminate and adopt those strategies in the laboratory. If you think about it, this is very cost effective. The only thing that might be needed to adopt this is a microscope, which laboratories already use for examinations, an Excel template, training of the personnel, and working together to a workflow mapping to see how that can be strategically be incorporated to assist in the analysis and minimizing bias in that examination. So with that, I would like to thank all the people that has made this work possible, the students, all the collaborators, the participants of the interlaboratory studies, and agencies that participated, and NIJ for funding. And with that, I will take any questions if there is time.
We have plenty of time for questions.
Excellent talk, as always. In all these comparison studies, would it be useful to use pattern recognition softwares in order to do it quantitative? And from software pattern recognition, you probably can evaluate all these probabilities, right?
Yes, correct. I didn't have the time to talk about all the things that we have done from a statistic perspective. But yes, we have used different approaches to calculate the score likelihood ratios, and we have created a Shiny app that is very easy to utilize, in which the examiner can input the score and they can see the distribution and see the score likelihood ratio depending on the subpopulation and the materials that are being investigated. We also did a very interesting collaboration with the physics department from a colleague of mine that is expert in computer science. So they collaborated in digitalizing all the database, and they use convolutional neural networks to do that comparison. And we have come up with different algorithms to do that in an automated manner and compare the output of the computer with the scores that we are reporting and complement that in the examination. We don't pretend to replace the examiners, but having a computer-based algorithm doing the comparison is very convenient because it helps us to determine whether or not we could have a potential bias if our scores are widely different. If they are not, that helps to support the examiner's opinion. Did that answer your question?
Yes, thank you.
Thank you.
I have one from one of our virtual participants. Who determines the minimum bin size for the ESS? For duct tape, it seems pretty straightforward, but for textiles or paint chips, it seems more ambiguous.
Yes, excellent question. I think that is one of the most important aspects that we have to define early on in the method development, and those decisions came across by discussing this with as many forensic practitioners that were providing us feedback. So the answer is as practical as possible, right? So in duct tapes, yes, we have the benefit that we have the systematic separation of the screen, so we use that in our benefit. For textiles, we have a relatively similar situation because textiles are constructed in such a way that has very common patterns that we can use to do the separation and establish what is the minimum size. But the size of textiles will depend on a case-to-case basis. So in that particular case, we decided to make a split of the width of the comparison of the tape into 10 bins. And we proceed to evaluate whether or not that was appropriate to capture those macroscopic and microscopic features at a level that can really demonstrate the thought process of the decision process of the examiner. For polymers, again, they can be large pieces or they can be small pieces that could come into a casework. So determine what was the minimum area. Again, came across to what are the features that we're evaluating on the microscopic level and how small we can go so that we can still capture very well those features that we are evaluating without making this examination too lengthy. So it was like a balance between practicality and ability to observe the relevant features still at the microscopic level. So yes, it's something that was not just very determined, but it came along with the collaboration with practitioners, feedback from them, and we came up with some criteria that is practical, functional, and has still been able to document the features that we are evaluating.
All right, thank you, Tatiana.
Thank you.
Our next speaker in this session is Igor Lednev. Igor is a distinguished professor in the Department of Chemistry at the University at Albany, SUNY. Igor has led long lines of research at NIJ applying Raman spectroscopy to forensic problems, including a body fluid ID line of research that is moved toward commercialization, and today he's going to talk about gunshot residue. Igor.
Thank you, Greg, for your kind introduction and for the opportunity to speak at this prestigious symposium. Today I will focus on the development of a new approach for gunshot residue detection and identification. This project has been supported by NIJ for several years, and I'm very grateful. And all opinions which I express today are of authors and not necessarily supported by Department of Justice. So in this presentation, first I, of course, introduce briefly the problem of gunshot residue, and I specifically focus on organic component, why it's important. And then we will talk about our new technology, which we are developing, and expansion of this technology to in situ measurements when we are trying to detect and identify gunshot residues on fabric without any any transfer of gunshot residue. And also, we will specifically look at size difference, especially on gunshot residue particles of smaller size. And I will finish briefly overviewing potential additional capabilities of this technology. As you may know, gunshot residue particles are formed when we fire the gun, and there are two major types, organic and inorganic. Organic particles are typically small, and they mainly come from primer, and organic particles are much larger, and they mainly come from smokeless, mainly come from propellant and smokeless gunpowder. Well, currently, the main method for identification of gunshot residue is based on scanning electron microscopy combined with energy dispersive X-ray spectrometry. And this method is based on identification of three elements, lead, antimony, and barium. And of course, with the development of environmentally friendly ammunition without lead, this method has some limitations. But the main reason why researchers are more and more interested in organic component of gunshot residue is because, first of all, organic particles are much larger, and they, by mass or by volume, organic component is also much larger and dominates gunshot residue which is formed. And at the moment, no false positives based on environmental contaminants are known for organic gunshot residue particles. And of course, it works for lead-free ammunition. And because of complex chemical composition of organic particles, many researchers are looking at the possibility to link organic particles to specific ammunition. There are several new technologies which are under development now for detection and identification of gunshot residue. Our previous speaker, Tatiana Travers, with colleagues, they are using LEAPS and electrophoresis for this problem. Also, DART mass spec has been reported can be used for gunshot residue identification, photoacoustic methods. But I'm a laser spectroscopist, so I would like to show you that spectroscopy actually has a great potential for detection and identification of gunshot residue because it provides molecular information, and potentially it should be confirmatory method for identification of gunshot residue. No sample preparation required. We are working on single particle level. Its method is non-destructive, inexpensive, single instrument. I will talk about this criterion of single instrument a little more later. And potentially instrument may be portable. Let me emphasize that gunshot residue detection is a twofold problem. First of all, we need to find gunshot residue particle on a relatively large area, on a relatively large surface, right? And then we need to confirm that the particle which is found is gunshot residue. And the current method, CMEDS, is two-step method. Scanning electron microscopy is used to find particles, and energy dispersive X-ray spectrometry to confirm that it's inorganic gunshot residue particle. Well, if we look at it from the viewpoint of spectroscopy, there are many instruments which already have optical imaging with software which allows to find particles as a first step, and several different methods which are used on second step. But I would suggest, of course, to use vibrational spectroscopy as a second step, because vibrational spectroscopy is well known as the most selective spectroscopic technique. It's perfect for identification purposes. And this is why we decided to combine fluorescence with Raman spectroscopy. And you see, we are using fluorescence instead of optical imaging, because fluorescence is known as the most sensitive spectroscopic technique, right? Detection of single molecule with fluorescence is not a problem. So detection of small gunshot residue particles, which have several fluorophores, is a relatively simple problem. And then, of course, Raman spectroscopy we are using because it's very selective. And what's important is that we can do it in a single instrument, combining these two techniques. I'm sure you all know about fluorescence, but let me briefly introduce Raman spectroscopy, because it's not as known as well as fluorescence. So we are using practically laser light, right, shining on the sample, and you see reflected and scattered light now, right? This is what we are practically collecting, and we are looking for photons which actually change energy. And this energy change due to excitation of molecular vibrations in the system. We analyze this energy change, we get what we call molecular vibrational signature of material. And as I already mentioned, this signature is considered as the most specific characteristic of the material. This work has been done in collaboration with Dr. Alexander Rzewski from Thermo Fisher Scientific. And this instrument which he provided for us can do fast fluorescent mapping and Raman spectroscopy. Since fluorescence is very sensitive, 2 milliseconds of measurement is sufficient in order to get fluorescent signal from different spots. So we just do mapping. And then when we find particle with fluorescence, we use Raman in order to map area around the particles. And again, single Raman spectrum, we are measuring using only 50 milliseconds. And this is typical result which we have. Here you can see particles on adhesive tape, which were found using fluorescence mapping. And then for each particle, we measure Raman spectra, and from those spectra, we identify whether it's a gunshot residue or it's environmental contaminant. And this is just representative of mapping, which we did with Raman spectroscopy, covering the area of the particle. And you see that we have very good chemical map, which can be used for identification of the particle. Recently, we moved forward from this experiment and did repeat it on cotton fabric. We practically use this cotton fabric as a target when we discharge firearm, and then we looked for gunshot residue particles on this cotton target using first fluorescence, and you can see we found quite a few particles. And then we did the Raman spectroscopy on individual particles, and again, we identified them as gunshot residue, mainly organic particles. And on this figure, you also see that there are spectra of Raman spectra of cotton, which are quite different relative to gunshot residue particles. All peaks in the spectra, we understand there is an assignment of these peaks based on literature data. and it's shown in the table. So practically, this means that we don't even need to use any transfer process in the beginning. We can look for gunshot residue particles immediately at the fabric. That instrument also allowed to do three-dimensional reconstruction using terrain approach. And you can see these three-dimensional figures, and you can see that particles are actually sort of embedded into fabric, and they are even partially covered with fabric fibers. Nevertheless, it doesn't prevent detection of these particles with fluorescence and identification with Raman spectroscopy. Well, these particles are relatively large. They are about 100 micron size, even a little larger. So we decided to look at smaller particles most recently, and it was done by my new student, Mackenzie Gascon. And she looked at particles size between 20 and 2 micron. And what she found is, first of all, she still get a very detailed spectrum, high-quality spectrum, Raman spectrum from each particle. And what was interesting is that all particles she investigated in this size range has contribution both from organic and inorganic components of gunshot residue. Well, it was sort of interesting for us, and it actually gives additional justification that this spectrum can be used as a very selective characteristic of gunshot residue, and will provide us confirmatory identification of gunshot residue. We plan to look at gunshot residue from other different types of ammunition to see if really in this size range particles are always a mixture of organic and inorganic components. It's interesting. And now let me very briefly talk about what other potential capabilities this method provides. Well, first of all, we looked at possibility to differentiate smokeless gunpowders using Raman spectroscopy between 3 different manufacturers. And for each manufacturer, 2 different ammunition types with different caliber. And in this paper, which we published in Hemosensus recently, we showed that we can actually identify all 6 ammunitions with 100% accuracy based on Raman spectroscopy. Why we would be interested in gunpowder? Well, combustion process during the firing the gun is incomplete, and it is well known that components of smokeless gunpowder are actually present in organic gunshot residue. So several laboratories around the world are looking for possibility to link gunshot residue to specific ammunition. And this is, I think, this is our goal too. And the first step is, of course, to show that, yes, at the level of gunpowder, we can differentiate ammunitions. So we need now look in details into the gunshot residue. And in fact, we already did some preliminary studies where we shot ammunitions with 2 different guns, 2 different ammunitions, and we used gunshot residue particles from 2 shots from each gun in order to build statistical model, and then we shot those guns again, take new particles, and with 100% probability on a single particle level, we identified from which gun that particle was created. So there is a potential possibility, of course, but more work needs to be done. And I also would like to mention this new approach which we started developing with Professor Baudelaire from Florida, where we combined LIBS, laser-induced background spectroscopy, with Raman spectroscopy, again, in one instrument. It is possible to do it in one instrument, right? And we did it in order to get both molecular and elemental chemical characteristics of gunshot residue. And we can combine these characteristics, and actually we get very high selectivity of this data set. In this particular work, just to prove the concept, we looked at gunshot residue generated from 3 types of bullets produced by the same manufacturer, right, with the same caliber, and all bullets were shot using the same gun. Nevertheless, with very high accuracy, we differentiated gunshot residue, which came from different types of bullets. I mentioned several times the one instrument. Why it's important that we are using one instrument? Well, for practical use, of course, it's important. You bring evidence sample and you run it in one instrument. Also, you need to use different methods. And as already mentioned, fluorescence microscope combined with Raman confocal microscope exists already, and we used it. LIBS combined with Raman also commercially available. And I don't see a problem, I mean, technical problem to combine all 3 methods in one instrument, fluorescence, LIBS, and Raman. And then it will be a very versatile method for characterizing, for first of all, fast detection, and then identification and even analysis of gunshot residue all in one instrument. And it's my last slide. I just want to invite you to my another talk, which I will give on Thursday. And this is on our attempt to use attenuated total reflection, Fourier transform infrared spectroscopy, which is also vibrational spectroscopy. And we want to use it as alternative we want to test it as alternative to Raman spectroscopy for identification of gunshot residue. Look at it. The instrument allows to get in 30 seconds over 1,000 spectra from different spots on the area of 40 by 40 micron. And on this figure, you can see a gunshot residue particle of 5 micron size. It turned out that it's inorganic, and we get full chemical characterization of this particle from a 30-second examination. So I think we have a great potential here. And our plans includes improving fluorescence characteristics, investigating potential environmental interferences, possibility to determine the time since the firing the gun, and expand method on different common substrates with potential fluorescence and Raman spectroscopic interferences. And of course, ultimate goal is to work on linking gunshot residue to specific ammunition and ammunition manufacturer. And I'm grateful to our collaborators and NIJ for funding and support. Thank you very much for your attention. I guess everything was clear.
Great work, Igor. Congratulations on the research. So my question is regarding the instrument of having the 3 techniques. Do you see that as a useful tool to do distance determinations, like shooting distance determinations?
Yes, and I see this as a great advantage. And what's also interesting is that this will be a universal tool, not only for gunshot residue. It can be practically applied for any trace evidence, right, including body fluid traces and hair and you name it. So you practically have very fast mapping capabilities using, for example, fluorescence. And there are very few examples of materials which do not fluoresce, right? And then you are combining LEAPS and Raman in order to get both elemental analysis and molecular composition of the sample, which gives you full information about chemical structure and chemical composition. So I think it should be very promising approach. Well, thank you very much for your attention.
Okay, so this is the final talk of this session. You'll see a theme here. So we've funded a kind of a burst in gunshot residue research recently. Our next speaker is Luis Arroyo, who is assistant professor in the Department of Forensic and Investigative Sciences at West Virginia University. Luis?
Thank you, Greg. So just one. Well, good morning, everyone, and thank you for the opportunity to speak today. Today, I'm going to be talking about transfer and persistence of inorganic and organic gunshot residues. Is the green one? Green button? Yep, I got it. Thank you. So first of all, I would like to acknowledge my grad students, or the grad students that actually are doing this work, especially Kearney, Thomas, Madison, and Leah. Also some of the undergraduates, students like Liana, Adio, and Ian, and also former students from our group, Colby, Corina, Bill, and Kearney. So without all their efforts, this work was impossible to achieve. Also, we would like to acknowledge NIJ for the support, the funding support. And also, I need to disclose that the opinions, findings, and conclusions are those from us and do not necessarily reflect those from the Department of Justice. GSR analysis is well established thanks to the confirmatory power of SEM-EDX. However, we still have some challenges, especially in the area of interpretation. So for instance, we can rely on additional tools to do a better job, especially if we can reduce the time of analysis from hours to minutes, and also if we can provide on-site testing to have better information, to better case management. So our research group has developed multiple tools, especially to build capacity. We are doing this via innovation, and we are creating fast and cost-effective methods that are compatible with universal collection tools, that are fast and provide accurate screening, also very versatile because they can be operated in the lab, but also in the field. And also they can be used in multiple applications and substrates. Also, the idea is that these methods are compatible with confirmatory tools like SEM-EDS and LCM-SMS analysis. So we can also conduct transfer and persistent studies. We can run authentic specimens and novel reference inorganic and organic reference standard materials and do this work on synthetic skin and use laser scattering for a purpose that we're going to study later during the presentation. Also, we have relied on the population studies and probabilistic interpretation, but today I'm going to concentrate mainly in the transfer and persistence due to time constraints. So this is our approach. We are trying to get a universal collection system where we can interrogate the inorganic components using LIPS and also using electrochemistry to do both inorganic and organic gunshot residues. Keep in mind that we can do this operation in less than 5 minutes. This is the typical workflow that we follow in authentic specimens. We collect using the carbon stop, the adhesive carbon stop. We can interrogate, let me see here. We can interrogate our specimen using LIPS to obtain the inorganic profile, but also later we can do electrochemistry, and because these two are non-destructive, we can conduct the analysis later using LCMS and SEM-EDS. So these are a list of the recent publications that we have published in recent times regarding our work in transfer and persistence since the year 2020 all the way to 2023 to give you some background information. With this, the idea is that we can plan on design a transfer and persistence study, and this is the way that we do it. We have developed and characterized standards both in inorganic and organic GSR. We have evaluated authentic skin from shooters and non-shooters, and we have created mock scenarios. And third, we have evaluated the synthetic skin models and developed comprehensive visualization tools, as we're going to see later during the presentation. So these plots over here, what it means is that we have found differences between the concentration levels between shooters and non-shooters. And in particular, I would like to raise your attention to, let me see here, the organic gunshot residue materials that are less prevalent than the inorganic ones as we have seen in our study. Also, because we're dealing with a large database, we can also do GSR population comparisons. The first comparison type that we did is the categorical, which is on the left in here, where we set up a threshold from the background population from which we can call it a positive. The second approach that we have conducted is the probabilistic outputs that are coming from the neural networks. And this with the intention to look into the the likelihood ratios of them. And as you can see on the plot on the right, on the right here, so for a shooter sample, we can obtain very good separation between the shooter after even conducting some post-shooting activities, differential from the low-risk population. Similar trends were observed in the case of the electrochemical work and LIPS, as you can see in these two plots. But the next step in innovation is, can we evaluate transfer and persistence under more controlled conditions? So first of all, we have developed and created a characterized GSR standards. In this case, we have the inorganic materials and the organic constituents. And therefore, keep in mind that we know in this particular case, we know the concentration of the compounds, we know the particle counts that are present, and we know that they are stable over time. So we know the ground truth, and this is going to be very important when we are going to be conducting studies of transfer and persistence, I'm going to be discussing later. So in this case, what we're going to do is, we have evaluated over 750 different samples coming from human skin volunteers and authentic shooters, and also synthetic skin and reference standards. We have simulated casework scenarios. We have evaluated the risk of loss, transfer, and contamination. And we evaluate several deposition mechanisms. So pay attention to this area because that's when we are looking into the synthetic skin. And keep in mind that we know beforehand the concentration and the particle counts at the time of the deposition in the synthetic skin. What we have done over here is that we deposit these materials into the synthetic skin. After we do that, we can collect the particles using the carbon stop. We conduct an extraction out, a step before here. We collect these particles and we evaluate the inorganic content using SEM-EDX. And then we perform an extraction out of the strat M material, and we can also evaluate this filtrate using LCM-SMS for the organic GSR content. And these are also some evaluation that we conducted in terms of the persistence, including actual shooter samples from human skin. In this case, we run the experiment between 0 to 3 hours. In the case of the synthetic skin, we extended up to 6 hours. And these are some of the major findings that we have found so far. In case of the organic residues, most compounds are detectable with minimum losses from human skin and synthetic skin for up to 3 hours. This is especially true when there's no rigorous activity after the deposition. The second major conclusion that we have get from here is that although some of the OGSRs are lost, you can observe that in the synthetic skin, after 6 hours, all compounds are still detectable. And the analyte loss, as we have learned from here, is also dependent on the physical chemical properties of the compounds. So in the case of the inorganic residues, we are able to detect characteristic and consistent particle GSR particles for up to 3 hours. Also, something interesting about this study is that the physical particles persist over time and are not significantly lost unless there's some sort of force that imposes on the particles themselves. So now we're going to move on to the actual activity and transfer and persistence using the same scheme model. In this case, we have a control of no activity, a set of 30 samples, and we're going to do some sort of activities that I'm going to describe here, like rubbing hands, right? We also using shaking hands and washing hands. And these are some of the studies and the trends that we have observed for the transfer of GSR and persistence. In the case of the OGSR, they are not really transferred from the primary surface to a secondary surface, and also the loss was observed during the activities depending on the rigor or extent of the activity, but there are no substantial transfer from rubbing hands or many of the other activities that we have performed. In the case of the inorganic residues, the secondary transfer was more likely compared to the OGSR. Even minimal contact, like a handshake, can transfer a number of particles. And more extensive activities, like washing hands, can lead to an almost complete loss of the particles. This is very relevant because we're going to assess now other type of scenarios. Do we observe similar trends in mock scene scenarios? So we would like to answer to this question. And in particular, we are interested to answer this one. Can we accurately detect GSR when there are potential transfer and persistent factors from the crime scene? So we assess this in 3 different scenarios. The scenario 1 is an arrest, scenario 2 is also an arrest, and a third scenario is suicide. I'm going to be describing the case scenario 1, and this is the workflow that we follow. Right now, we're using a mobile LIPS device to obtain the inorganic analysis. We use electrochemistry also in a portable device to do the same work, plus the organic GSR. And finally, we obtain the confirmatory analysis of both using the LCM-SMS system. And this is the question of interest. Is there any GSR transfer during the handling of the firearm? The second question that we may ask is, if a suspect only handles a firearm without shooting, if there is GSR secondary transfer to the arresting officer. And this is the way that the dynamic operates. So first we have a suspect who handles the firearm for a minute and then wait for 15 minutes. Second, an officer handles a firearm and then washes the hands and drives for 15 minutes. And then later, the officer arrests the suspect. And then the idea is to respond to the questions of interest that I highlighted over here. So these are some of the major conclusions that we obtained from this scenario. And the first one is that the inorganic GSR transferred and persisted on the individual who only handled the gun without firing. You see on the table below that no organic was detected by any of the methods using the firearm handling, and no GSR secondary transfer was observed from the person of interest who handled the firearm to the arresting officer. And washing the hands after handling the gun removed GSR particles. So these are interesting areas that we have concluded for this particular study. So what else can we do to understand the deposits of inorganic gunshot residue and organic gunshot residue deposits? And that moves around. What else can we do about this? And therefore, the purpose of the next study is to develop methods that can help us to visualize and understand the flow and deposition trends of the GSR. And in this case, these are the three major questions of interest. The first one that we want to address is what GSR sizes and distributions are observed during an event. What about the deposition and settling of the GSR particles? And finally, try to understand the risk of exposure to GSR by the shooter, bystander, and passerby. And this is when innovation comes into play. So the experimental approach that we follow has several layers of information. First, we would like to see, can we see and visualize the particles that we're dealing with? And for this, we're using laser sheet scanning for this purpose and also high-speed videography. The second layer of information has to do with the particles. Those particles that we are able to see now, can we measure them? And we use an aerodynamic particle sizer and also simple and cost-effective particle counters. And finally, can we analyze those particles? And the answer is regarding the use of an SEM and LCM-SMS instrument. And the answer is, of course, yes. So I'm going to raise your attention to this video that now facilitate our information. Now we can see the particles. We can see the plume of the GSR happening after a firing event that before we couldn't. So this is an important component. And now that we can see those particles, can we count them? And we develop and strategically move and position these sensors. And I'm going to describe the sensors in a minute, but also I'm going to show you here the position of the sensor. So this is we're in the close vicinity of the shooter, and then in the passerby all the way in the back, and also the person next to the shooter. And also important to highlight in here is the actual counters. So these are passive counters that are going to be positioned, as I mentioned, 3 in a row in each of these positions in the shooting range. And something that we need to consider and guarantee is that there is no cross-contamination or signatures from the shootings in between the experiments. So in this case, we are using the ventilation in the shooting range in order to avoid this effect. And the help of the visualization helps us to guarantee that aspect. And another area of interest for us was to understand risks of deposition. And this time, what we did is to flip the laser sheet. Instead of being vertically positioned, now it's horizontally positioned. And as you can see in this video, right now we can observe that the person, the bystander person that is next to, in this case, of the shooter, and you can see the reach out of the particles. And we can now, using this analyzer, we can count and found similar numbers of particles in between the shooter and the bystander. So there are risks from contamination from the firearm in these two scenarios. And also, we can quantify the number of particles using these passive deposition stops. So what you can see is that we have a comparison between characteristics and consistent particles, right? So this is the numbers, excuse me for this, to get back. So these are the numbers of particles that were found in the vicinity of the shooter, but a significant number of particles were also found in the bystander and the passerby individuals too. And remember that now we also collect and stop the hands of the shooters, the hands of the bystanders, and the passerbys. And something interesting that we found is by doing this analysis, we observed differences in between them by particularly the organic gunshot residue deposition. And the second aspect that we found is that the organic gunshot residue deposition decreased within decreasing distance from the firearm. So just to wrap up, our preliminary findings in here, the GSR dynamics and exposure, we know that the GSR spreads greater than 14 feet through the enclosed space in less than 5 minutes. The data indicate that an estimated GSR setting times may be temperature dependent at this stage. And the analysis of the video and the particles indicates a risk of exposure to bystanders and passerbys, especially for the inorganic GSR. Also, monitoring the OGSR deposition on the hands indicate less risk for bystanders and passerby. And finally, probably the most important aspect is that the data indicates that the combination of inorganic and organic GSR analysis may provide further interpretation of shooter versus bystander versus passerby. And two more conclusions in here. The inorganic and organic gunshot residue mechanism for deposition, transfer, and persistence have significant differences that we need to understand to evaluate the evidence under a broader context. And secondly, the combined use of the inorganic and organic GSR data and increase our knowledge on transfer and persistence can lead to more comprehensive interpretation approaches. And with that, I would also like to thank some collaborators, NIJ again for the funding, and some collaborators from the University of Auckland, particularly James Curran from NIST, Matt Steinmetz, who is also a co-author in this presentation, and SELEC and the University of Costa Rica and some other applied spectra for helping us with the development of the LIPS, portable LIPS unit, Metron, and some collaborators from the Sacramento State Police and New Jersey State Police and Iowa State University, and of course our department for facilitating the resources for the shooting range. And with that, I hope that you have a good rest of the morning. Thank you. We're ready for a break.
Thank you. That was a very interesting talk. I'm not as familiar with organic gunshot residue, but you mentioned its stability. And I was wondering, in terms of timetable, how stable is the solution that you've created? Like, when do you start to see degradation?
That's a very good point. And actually, we conduct experiments of aging, if you will, on this solution up to 12 months. and we haven't noticed any differences in the concentration, and that's part of the success of this particular standard that we created. And we haven't observed any degradation at all in the system, in the standard. Question over there.
Luis, I liked watching those videos. I had a question. So it seemed like the bystander is that the person standing next to the shooter. It seems to me, from the video at least, that there would be really no difference between the number of particles that would appear on someone standing next to the shooter.
Yes.
The actual shooter. It seemed to me that there was so many. Right? And then you said that it went down by distance. I was just wondering what kind of distance you would.
Yeah, we have in the shooting range particular, it's a trailer basically, so we have set up the counters at different distances in between. Not only consider the shooter, which is actually doing this, so we have the counters in this region, and also the bystander. So based on those informations and the visual aids that you observe, we arrive to those. So as long as the person moves back in this enclosed, remember there is an enclosed environment, the distance has a role in determining the amount of particles that are going to be reaching the individual. That's exactly why we are trying to do these studies, because we need to understand not only the dynamics of the inorganic material, but also the organic ones, which is a critical component. So they tend, and you observe in the table that I present very quickly probably, but then there are differences in the behavior for the organics versus the inorganic ones.
I have a question from our virtual attendees. They asked, tests were carried out indoors. How much will be the variation outdoors? Tests were carried out indoors. How much variation?
Yeah. Well, of course, outdoors is a pending task for us, and we already have a plan lined up to do these studies in an outdoor scenario. And the person is right. It is not the same to be in an indoor facility that versus an outdoor facility. So these results are, of course, under controlled circumstances. But we already have a plan lined up for this summer to conduct outdoor activities, so that maybe next meeting I can give you some answer for that.
All right, thank you, Luis.
Thank you.
So that concludes the first session. We will take a short break and reconvene at 10:35 for forensic anthropology and pathology.